Hello and welcome back to Bioclass Bytes. In this video, we are going to talk about theories on the origin of life. Don't forget to subscribe to my channel and like and share this video. These are the unit and lesson titles for this video playlist, and in this video, we are going to focus on theories about the origin of life, the chemical evolution, and the Miller-Urey experiment. Before you continue with the rest of this video, I recommend that you watch this one first from Stated Clearly, entitled Can Science Explain the Origin of Life? This is a very informative video. All the concepts are related to our discussion today. I'll provide the link in the description below. The question of how life originated is one of the biggest questions, okay, that scientists are still trying to answer. It's quite difficult because, number one, it's impossible to go back in time. We do not have the technology yet to time travel and observe life's beginnings, okay? Nor are there any witnesses to actually tell us what really happened. However, despite of those challenges, what we have are pieces of evidence surrounding us. Okay? And these pieces of evidence could give us an idea on what really transpired millions and billions of years ago. So what scientists are doing is to gather all of these pieces of evidence and try to um, come up with hypotheses or theories that could possibly unify all the existing data that we have. Okay? Connect in relation to this question on the origin of life. So there is testimony in the rocks of the earth. We have fossil records. We have information uh, s- even on the rock layers that are exposed right now. Ice cores that we've drilled through ice sheets. Okay. However, this evidence is not easily read, and it is often silent on issues. But it is the task of science and scientists to actually figure out what they mean. Now, with regard to different theories about the origin of life, we actually have a lot of theories. Okay, um, for it is actually natural for our species to be curious about how things came to be. That's why I'm pretty sure you know about this. That every distinct culture in every span of human history have their own creation stories. Ancient Egyptians have their own creation stories. Ancient Greeks, Japanese, Chinese, um, different races and different tribes and different cultures. Even our own here in the Philippines, we have our own creation stories. Um, for the purpose of discussion, we will narrow down the theories into these four: special creation, spontaneous generation, extraterrestrial origin, and abiogenesis and biogenesis. We've just mentioned that there is testimony within rocks, and this one is a gallery of different fossil types that we have uncovered so far. So we were able to find fossils embedded in sedimentary rocks. As you can see here, a plant fossil about 300 million years ago. Okay. Then we also were able to discover petrified trees about 215 million years ago, and they are considered mineralized organic matter. Okay. Minerals seep into and replace the organic matter in this tree. Then we also have trace fossils, okay, of footprints, burrows, and other traces. As we can see here, 150 million year old dinosaur tracks in Colorado. Imagine how big, okay? We also have amber. These are entire organisms preserved in hardened resin from a primitive tree. Okay, around. So this one is an insect in amber, about 40 million years old. And then we also have frozen soil, ice, and acid bogs. Okay, those are good substrates for preserving. In this case, a 50,000-year-old wolf pup found in frozen soil in Yukon, Canada. Okay. Um, So these are all the evidence that we keep on discovering, and what scientists do is to analyze and research about all these fossil evidence, and then try to fit them into our history. The theory that life was created by a divine being is the core of most major religions. Okay, it's the special creation theory. It is the oldest hypothesis, okay, about life's origin, and for the longest time, it is also the most widely accepted. When I say widely accepted, in terms of human history, okay, um, before the dawn of scientific revolution in the past, human beings have tried to explain the origin of different things, and most of the time they attribute it to supernatural or divine um, origins or divine explanations. Some For Judeo-Christian believers, they believe that God created life, um, and that they believe it more than other hypotheses. And many have taken an extreme position, and by proposing or by professing that the book of Genesis from the Bible actually happened and are factually correct. Okay. Um, so according to the book of Genesis, God created everything within 6 days. The day and night, atmosphere, dry land and plants, sun, moon, and stars in this order, sea and flying creatures on day 5, and then land animals and man on the 6th day, and on the 7th day he rested. This is one of the widely accepted creation theories. Let me share this article printed uh, on Catholic Herald. It was written by Patrick Cosworth in October 31, 2014. The title says, Pope Francis's comment on the Big Bang are not revolutionary. Catholic teaching has long professed the likelihood of human evolution. If you want to read this article, I'll provide the link in the description below. Now, these are the important parts of the article, okay? In his address to the Pontifical Academy of Sciences, Pope Francis warned against the image of God, the Creator, as a magician with magic wand, okay? He also said that belief in both theories about the origins of the universe, okay, so that would be the Big Bang, and the birth of humankind, or the rise of the humankind through natural selection, through evolution, are all consistent with Catholic faith. Now that's quite a refreshing comment. That's quite a refreshing um, statement coming from the sitting Pope. He also said that the Big Bang, which is today's most widely accepted scientific explanation about the origin of the world, does not contradict the divine act of creation, rather requires it. Okay? He also said that evolution of nature is not inconsistent with the notion of creation. Okay? Because evolution presupposes the creation of beings which are capable of evolving. So these are very refreshing take coming from the sitting Pope. And this was that address, okay, that they mentioned, okay? You can watch this video. It is translated in English. It has a subtitle, so you can watch it comfortably. The title of the video says, Pope Francis, Big Bang and Evolution Confirm God Exists, okay? So I recommend, okay, that you watch this video and see where the current Pope is coming from, and see that there's really not this huge divide between religion and science when it comes to the origin of life on our planet. Now, I also recommend that you watch this video from TED-Ed entitled The Five Major World Religions. Um, so just for us to have this perspective that Catholicism, Christianity is one of the five major religions, and each major religion also has its own creation story, special creation story, and it will do us good to actually take the time and listen and learn more about other religions, just so to end the stigma, this stigma or this idea of separation, and discrimination from each other. Okay? So watch this video. Let's enrich what we know about other religions and what they say about creation. I'll provide the link in the description below. The next theory that we will talk about is spontaneous generation. Now, before we go any further, I want you to know that this is actually an obsolete theory. This has already been disproven. Okay? But just to see the historical development of the concept of life, I decided to include this in our discussion. So, spontaneous generation, spontaneous, instantly, okay? Right away, okay? Generation or creation, okay? Spontaneous generation refers to the formation of living things without descent or without coming from similar organisms, without having descended from parents. Okay, so people in the past, before scientific revolution, before enlightenment, once believed that mice come from corn, flies come from bovine manure, okay? Maggots come from rotting meat, and fish from the mud of previously dry lakes. So they even had recipes for organisms, such as this one, recipe for bees. That is to kill a bull during the first thaw of winter, build a shed, place the bull and herbs and all that. Then when summer comes, they will find a decaying body of the bull with bees. Okay, so back in the day, this is what scientists, respected scientists, naturalists, and philosophers thought about the origin of life or how life came to be. Okay? But again, no, I want to emphasize spontaneous generation has already been disproven. It is already considered an obsolete body of thought. Crucial to spontaneous generation is this, that life comes from non-living um, materials, that life comes from non-life, and there's no causal agent, nothing triggers it or nothing causes it, such as the parent. Okay? Now, the doctrine of spontaneous generation was synthesized by Aristotle. I think you're familiar with Aristotle, no? He compiled and expanded the work of earlier natural philosophers and other various explanations. Okay, and spontaneous generation was accepted as a scientific fact for 2 millennia. That's quite a long time, 2,000 years. Okay, it remained unchallenged. However, with the advancement in science and more people taking interest in this field of study, um, of course, spontaneous generation will eventually be challenged. Okay, there are a lot of loopholes in this theory, and it was only natural for scientists to actually take notice. Now, the first major challenge against spontaneous generation was conducted by Francesco Redi in 1668. He was referred to as the founder of experimental biology. Okay? He was the first person to challenge the theory of spontaneous generation by demonstrating that maggots are actually they actually came from eggs of flies. Okay? So this was his 1668 experiment. Okay? So he had wide mouth jars containing with a piece of meat. One, an open jar, okay, with the meat inside. Another jar but it's covered with gauze. Okay? And he had another jar but sealed or covered. So he observed what happened after several days, and this was his findings. The open jar, the open jar with the meat inside, eventually developed maggots because flies were able to enter the jar and then lay eggs on the meat. Then eventually the eggs hatch, and those are the maggots. In the gauze-covered jar, okay, no flies were able to enter the jar, but they were able to lay eggs on top of the gauze. Okay, and some of the eggs possibly fell through the gauze and then landed on the meat. Then eventually, maggots still hatched from the meat. His third setup was a completely sealed jar, so no flies were able to enter or and then lay their eggs. Therefore, no maggots or eggs, no maggots were developed from those. Okay, so this led him to conclude that if there are no flies entering the jar, then no maggots will form because the maggots are actually the offspring of those flies. Okay. This was a challenge to spontaneous generation, which tells us that life can come from non-living matter, that maggots will come from this non-living meat. But actually, what he was able to prove is that if the flies cannot enter and lay eggs, no maggots will develop. So this was quite a revolutionary proposal, revolutionary findings, um, and one of the first major challenge against spontaneous generation. People were still unconvinced with Redis results, okay, and it created this back and forth um, communication between scientists as they try to prove or disprove spontaneous generation. Now, the next major event came from John Turberville Needham's experiment in 1745. Okay, so Needham was an English biologist and Roman Catholic priest. He did experiments with gravy. This time he boiled the meat and then he used the broth as his experimental setup and tainted wheat and containers. Okay, so what he was trying to prove was spontaneous generation is true. Now these are his experimental setups. The first one was he placed the broth in this flask, boiled the flask, let it remain open, and after several days, I was able to find and see microorganisms in the broth. His second setup was this. He boiled the flask with the broth inside and then covered it. Then after several days, he was able to find microorganisms inside. This led him to conclude that spontaneous generation is true, that microorganisms could actually arise, could actually come from the non-living broth. Okay? Because in both setups, if perhaps life essence could come in and then reproduce in the open flask, but how about those covered flasks? How come that at the end of the observation period, microorganisms still formed in the closed, okay, or sealed flask? Now, the only criticism, okay, of Needham's experiment was this, okay. This must be because the cork and the flask were not sterile. They were not sterilized or boiled or cleaned. Okay, they were not devoid of already contaminants. Okay, they might have contaminated the broth inside, bringing in microorganisms which eventually reproduce with the nutritious broth. Okay, so this was in support of spontaneous generation, but people were still, not fully convinced. There were still a lot of back and forth between scientists in 1745. The next experiment came from Lazzaro Spallanzani in 1767. He was an Italian Catholic priest, biologist, and physiologist. Now, his research, okay, of biogenesis paved the way for the downfall of pre-formationism theory, or the idea that organisms develop from miniature versions of themselves. Now, this was his experimental design. So he still used broth the same way as Needham, okay? So he heated the broth, left it open, then after several days, he was able to find microbial growth in it inside, okay? His second setup was this. He heated the broth inside, then sealed it. And after several days, he was not able to find any microbial growth. This time, he was able to actually observe that no growth developed from the sealed flask. Only when he opened the flask did he see microbial growth inside in the broth. So this led him to conclude that tiny organisms from the air possibly caused the growth in flask A. Okay? And they were not able to enter the covered flask in setup B, so therefore no growth were seen in flask B. Only when the flask were open did microbial growth were seen in the broth. Okay, so people were hoping that this would have ended the debate on spontaneous generation, but the supporters of spontaneous generation argued that possibly he boiled the broth in setup B too long and must have probably killed the essence in the broth. Okay? So this was still unsettled even after 1767. Now, the experiment which ended the debate was conducted by Louis Pasteur in 1859. He was a French chemist and microbiologist, and he was best known for his technique of treating milk and wine to stop bacterial contamination, which was eventually called pasteurization to honor him. Now, this was his experimental setup, okay? He still used broth, okay? But he had three setups. The first one was um, here, the broth was boiled for 1 hour to sterilize it. If you still remember, that was the criticism of Needham's work as well as Spallanzani's work. So all of the flasks were boiled for 1 hour, okay? And then allowed to cool. His first setup had a flask with an open neck. Okay? This allowed air and bacteria to enter, and as expected, after several days, contamination was visible. Okay? The broth was contaminated with bacterial growth. His second setup was the same, except that instead of being an open jar, the neck was filtered, okay, or was plugged with cotton, which filtered the bacteria. Air could still enter, but bacteria could not. After several days, the broth remained sterile. No bacterial growth was observed. Okay, so this is one way already, okay, that to disprove spontaneous generation. But Louis Pasteur took it even further. He created another type of flask, okay, with a swan-like neck, like this, which was kept open at the end. Okay, not just like this one, but it has a swan-like neck. Now, this was a good idea because it remained open, allowing air to enter, but not bacteria. Okay? Bacteria were removed from the air entering the flask. Okay? And they were only found settled on near the opening, near the tip of the neck. Okay? And as expected, even if it was open, the bacteria cannot go in. The broth remains sterile even after several days. No contamination. Only when Louis Pasteur tilted the flask, tilted so that the broth will reach the tip, will reach the bacteria in the opening, only then were they able to find microbial growth. After several days, no, microbial growth in the broth inside. Now, in this setup, people no longer had anything to say because it was open. All of the setups were boiled for the same period. All of them were sterilized. All of them were allowed to cool down. In this part, the broth was exposed to the air but not to the bacteria, which are actually considered as the parents of whatever contamination you will find in the broth. This finally ended the debate, finally disproving spontaneous generation, that it is not possible for life to come from non-living objects, non-living materials. Today, Pasteur is often regarded as the father of germ theory. Okay? The theory that states that germs or pathogenic organisms are the ones who cause diseases. Okay? He was also the father of bacteriology because of his experiments. Okay? It was the study of bacteria, and he founded it together with Robert Koch. So indeed, Pasteur was able to show that the growth of bacteria in nutrient broths is not due to spontaneous generation, but rather to biogenesis. Okay? This term means life, bio, genesis, creation. Okay? Life coming from life, or living organisms only coming from other living organisms. If you actually go to Science Museum in London, you can still find um, one of his bottles, okay, one of his flasks, okay, that was sealed before, okay. He sealed the flask like this one, and no air was able to enter the flask. Since 1859 up to today, his broths remain sterile, okay, and are still available for viewing. So, since air and bacteria cannot enter, no matter how long it has, no matter how much time has passed already, his broths remain sterile, free from contamination. He further showed that fermentation, okay, is caused by the growth of microorganisms, okay, in his work. And finally, he showed that pasteurization, or the heating of liquids to a certain point in time, sterilizes them and allows for long-term storage. And back in the day, that was actually a huge deal, the ability to preserve food for longer. And as I've mentioned, the process was called pasteurization to honor Louis Pasteur. If you're interested to learn more about Louis Pasteur, I recommend that you watch this video entitled Louis Pasteur Scientist. I'll provide the link in the description below. Extraterrestrial origin is another um, theory on how life exists on our planet. So this time here, extraterrestrial, it means that the origin of life came outside the Earth, or outside our planet. So this is also known as the theory of panspermia, which proposes that meteors and cosmic dust that used to hit primordial Earth, or used to hit our planet in the past, may have carried significant amounts of complex organic molecules to our planet. So when an asteroid hit our planet in the past, billions of years in the past, or when cosmic dust or meteors hit our planet, they might have carried very important molecules that were ingredients in the early cells or early living organisms. So hundreds and thousands of meteorites are known to have slammed the early Earth. They may have carried those organic materials. So the theory of extraterrestrial origin or panspermia tells us that in the primitive Earth, cosmic dust, possibly, for example, from Mars, when it was hit by asteroids, so cosmic dust coming from that event might have reached our planet, or when we were constantly bombarded by asteroids or meteors, and they hit our planet, early planet all the time. So these are possible carriers of building blocks of life, or organic materials that were necessary to kickstart the evolution of life on our planet. So they might have hit our primordial ocean, or they might have reached those hydrothermal sea vents, okay, um, under our primitive ocean. So the chemical materials coming from outside the Earth might have interacted with the physical events going on with our own planet, with our own primordial planet. And that might have possibly be the cause of the evolution of life on our planet. Okay? Or some of those cosmic dust or organic materials might have reached the tidal flat. Oh, so that's where that's where bodies of or landforms interact with the bodies of water due to the rise and fall of the tides. So there must have been some chemical interaction in this physical environment here. So the theory of panspermia tells us that the building blocks of early life might have come from outside our planet. So when they entered our planet and interacted with the physical events that's happening in the early Earth, that might have caused the early life forms to evolve. So again, this is what theory of panspermia, or the neo-panspermia model, proposes, that meteors or cosmic dust deliver these building blocks for life, for generating life, after the formation of the Earth's ocean and atmosphere. And they are still doing that. These are still happening in the world today. So meteor delivers building blocks of life in bodies of water, or in bodies of land, or bodies of water, and that's where the chemical processes originated. And then together with the UV radiation, all of those were actual ingredients of the early life forms. Finally, we have abiogenesis and biogenesis. Okay? So abiogenesis is the natural process of life arising from non-living matter, such as simple organic compounds. Now, do not confuse this with spontaneous generation. What spontaneous generation tells us is that living organisms, an organism right away, will come from non-living matter. What abiogenesis is telling us is the historical process of life coming from simple organic compounds. Okay? So this was the history of how life happened, not just of how an organism is created. So abiogenesis, according to science, is thought to have occurred on Earth between 3.8 and 4.1 billion years ago. Now, this is the simplified theory of abiogenesis. It says that simple chemicals that are present in the Earth's environment back then, okay, Earth's primitive environment, okay, must have been triggered with physical events that allowed them to form polymers or longer chains of molecules. Now, these polymers eventually gain the ability to replicate themselves and to undergo that continuous cycle of replicating and creating and replicating themselves up to such a point that they were able to create a protobiont or a primitive cell. That primitive cell must have undergone through natural selection within that primitive Earth's environment to eventually evolve and become our what we consider a living thing as bacteria. Now, this simple process of abiogenesis, again, do not confuse this with spontaneous generation. Abiogenesis tells us the history, the possible history of life. Spontaneous generation refers to the creation of just a single organism. Now, abiogenesis is studied through a combination of laboratory experiments and extrapolation from genetic information of modern organisms in order to make reasonable conjectures, okay, about what before living organisms, when there were just simple molecules, pre-life chemical reactions may have given rise to a living system. So this is scientific. Abiogenesis is a scientific explanation. Okay, that is continuously being tested and studied as we speak. Um, Nonetheless, no, Earth is the only place in the universe known to harbor life. This process of abiogenesis, even if it is simple, there's a possibility that this could have also occurred in other planets, that are similar to Earth's primitive environment. However, no, Earth is the only place that is confirmed to have undergone such process. Um, because of this, one of the researchers said that if life arose relatively quickly, relatively in terms of millions and billions of years, okay? If life arose relatively quickly on Earth, then it is also possible that it's common all over the universe. And that is why we need to explore other planets in our solar system and then eventually gain the technology to go out and leave solar system and explore further into the stars. Now, this is the life timeline, okay, of how life possibly originated. So we have the earliest Earth, okay, in a scale of millions of years, 4,500 or 4.5 billion years ago. Then we have this primitive water Earth. Okay, we've mentioned that already in one of our previous videos. Now, earliest life possibly occurred 4.1 billion years ago. Okay? In those primitive waters. In those, if you still remember, no, we've mentioned that the origin of life is possibly due to the combination of physical events and chemical processes that happen in the primitive Earth. So sometime here, somewhere here, okay, wherein there were just molecules, life took a leap, and the single-celled organism was born or created or evolved during this period 4 billion years ago. And that's what scientists are trying to look for. Then eventually, the single-celled life continues to evolve. So we now have photosynthetic microorganisms, which gave rise to eukaryotes. Eukaryotes eventually evolved into different multicellular organisms, and were able to colonize the land. Then we have dinosaurs, mammals, flowering plants, and now earliest humans or human species. So if you notice, our species is actually relatively young compared to the rest of the living things, and even compared to the rest of the planet. Our species, Homo sapiens, has only been around for approximately 200 to 300,000 1,000 years ago. And we're talking about billions of years here in retrospect. Okay? Now, this diagram shows us how abiogenesis could have possibly occurred. Now, this one was data collected, okay, of features of abiotically produced vesicles. So, vesicles are what we think of as the primitive cell membrane, okay? Vesicles like this possibly are the first type of cell membrane. So, they noticed that in the presence of morillionite clay, so that's a type of clay, type of soil, greatly increases the rate of vesicle self-assembly. They noticed that if you actually introduce, so if these are all the molecules, okay, non-living molecules, just molecular polymers, all of this, if they were introduced to the presence of morillionite clay, the rate of chances, okay, the rate of them forming their own vesicle increases over time, even if we're just talking here about minutes, since we are just looking at molecules. So in a span of less than 20 minutes onwards, okay, the precursor molecules plus the morillionite clay were able to assemble their own vesicles. Okay? So that's the first thing. And this is probably what happened in the primitive Earth. Our protobionts were eventually able to have this vesicle to separate them from the world around them. Not only that, vesicles were able to divide on their own or create smaller vesicles. As we see here, a vesicle giving birth, quote unquote, to smaller vesicles, okay, after being exposed to morillionite clay. And finally, the third step, they were able to absorb RNA or ribonucleic acid, which is actually a subunit in organelles that are in charge of photosynthesis. So how did scientists know that they were able to absorb RNA? They actually coated RNA in this orange pigment, okay? And they were able to see, so they used to be floating here around, and then the vesicles were able to actually absorb that RNA and keep it inside their boundaries. So possibly we are looking now at how those protobionts were formed, how those protobionts eventually evolved and become our first bacteria. What we've previously mentioned was abiogenesis, the possible rise of protobionts from molecules, okay, through the events that we've mentioned. Now, what happened after? Happened after this? What happened after this hurdle? We call that biogenesis. So this is the production of new living organisms or organelles. So as you notice, the a here, the prefix a, which means non-living, is removed because now we are talking about the production of of living organisms, creation of living organisms. So this is abiogenesis, also known as chemical evolution. So this is what I was telling you about, early Earth. Then you have your inorganic chemicals, okay, polymerization, eventually vesicle formation, okay, the primitive form of plasma membrane. When they made that jump and become the first type of cells, this is the start of biological evolution. This is where biogenesis now happens. So the law of biogenesis attributed to Louis Pasteur is the conclusion that complex living organisms, so living organisms can only come from other living organisms. So cells can only give rise to other cells. Okay? Living organisms can only give rise to living organisms. This was proven by Louis Pasteur in his experiment. And that process is called reproduction. Okay? So here, if you notice, this is just the reproduction of, molecules. We call it polymerization. But once they cross that threshold, it happened 4.1 billion years ago. Once they crossed that threshold and they started to exhibit the characteristics of life, when they reproduce, it's now called reproduction, no longer polymerization. Okay? So this is summarized in the phrase omne vivum ex vivo, which means all life from life. Okay? So this is also a related statement of omne cellula e cellula, which is actually one of the central statements of cell theory, which states that all cells can only come from pre-existing cells. So again, abiogenesis, that's chemical evolution. It tells us how life possibly originated during the primitive Earth from these organic molecules. Then as they cross over, they evolve into living organisms, and now this falls under biogenesis or biological evolution. Now, after that, biologists attempt to classify organisms in different categories based on the characteristics that they exhibit. So we have what we call the three domains of life. So this is the common ancestor of all living things. And as time goes by, okay, as billions and millions of years went on, eventually they split apart from each other, forming the domain bacteria, okay, what we know of. Then domain archaea, so these are prokaryotic unicellular organisms who lived in extreme environment. And then domain eukarya, so these are eukaryotic organisms. Okay. Then this one is a more elaborate tree of life, okay, that shows us, okay, and it is still continuously being revised, okay, as we keep on discovering more and more organisms. So our common ancestor of all living things, we have a name for it. It's called LUCA, L-U-C-A, last universal common ancestor, which eventually divided into your domain bacteria, okay, and these are all the groups of bacteria underneath it. Domain archaea, and these are the groups, smaller categories under domain archaea, and then domain eukarya. These are the rest of the organisms. So you have here your algae, fungi, you have your animals and plants under domain eukarya. This is another table now showing us the three domains of life. So you have your domain bacteria. These are the most diverse and widespread prokaryotes. Okay? Then domain archaea includes multiple kingdoms. The thing that separates them from the rest of the domains is that they live in extreme environments. Okay? Such as salty lakes, boiling hot springs, and others. Okay? Now, the rest of the organisms that we are familiar with are under domain eukarya. They are eukaryotic organisms. You have kingdom plantae here, photosynthetic organisms, saprophytic fungi. You have animals under kingdom animalia, and protists, which are mostly unicellular eukaryotes. Now, we've mentioned chemical evolution, okay, a while ago, okay, under abiogenesis. Now, to understand that further, to be explained deeper for you, I require that you watch this video from Stated Clearly entitled, What is Chemical Evolution? It tells you everything you need to know about abiogenesis leading to biogenesis, and I'll provide the link in the description below. Now, whenever we mention the chemical evolution, we have to talk about the Miller-Urey experiment, okay? So, Miller-Urey are two scientists, okay? So, this is Stanley Lloyd Miller, and this is Harold Clayton Urey. So, Miller-Urey, okay? It was a chemical experiment in which these two scientists, tried to simulate the conditions of the early Earth. Okay? They try to simulate, they try to recreate the conditions of the early Earth and tried to test the chemical origin of life under these conditions. They tried to recreate the physical environment. Now they're looking at the chemical processes and see if it is possible for life to come from it. This was considered to be a classic experiment investigating abiogenesis. It was conducted in 1952 by Miller and Urey at the University of Chicago, and it was eventually published the following year. The experiment, Miller-Urey experiment, confirmed what Alexander Oparin, shown here, and J.B.S. Haldane said that the conditions of the primitive Earth favored chemical reactions that synthesize complex organic compounds from inorganic precursors. Again, this term keeps on coming up. Physical events plus chemical processes. The perfect combination of Earth's primitive environment with chemical processes are possibly the source of life on our planet. It shows us how from simple inorganic molecules they evolve and become the precursors of life, or our protobionts, or our protocells. Stated Clearly has a dedicated video just for the Miller-Urey experiment, so I recommend that you watch this one. As usual, take down notes of important concepts. I'll provide the link in the description below. Now, this was what Miller and Urey were trying to recreate in their experiment, the physical environment of the primitive Earth. Okay, so they were considering carbon dioxide in the atmosphere, which combines water from the clouds, and then eventually carbonic acid was formed, which reacted with the rocks there. Then those molecules were carried into the oceans, and calcium and bicarbonate form calcium carbonate, okay, after precipitation. So this is what they were trying to, one of the things that we're trying to look into as one of the conditions of the primitive Earth. Now, this was their experimental design. This was actually their apparatus, okay? It was consisted with a closed tube connecting two chambers. So, closed tubes connecting two chambers. The upper chamber is actually a simulation of the primitive Earth's atmosphere. That was what they were trying to recreate. So, they put all these chemical compounds, these gases, into this gas mixture. Okay, that's one. And they also placed electrodes to discharge sparks to simulate or to copy lightning. Okay, so the other chamber tried to recreate Earth's waters. Okay, Earth's hydrosphere. Okay, so that was the idea. Okay, so what they did was to actually heat the water or heat the oceans. Okay, heat source, possibly coming from those hydrothermal vents from the magma chamber, and then as water boils, okay, of course it evaporates. Okay, and then it goes into the gas chamber. Okay, wherein I've mentioned a while ago they were trying to recreate Earth's primitive atmosphere. Okay, now in the condenser, okay, the water vapor cools, okay, causing water droplets to form. Now this condenser eventually allows condensed liquid mixed now with complex molecules to go down and settle into this. Okay, um, they will be dissolved in the droplets and carried into the ocean chamber. Okay, for which samples were withdrawn for analysis. Now, this water will now return to the boiler, and then this cycle was repeated during 1 week. So this is what they try to do: boil the water, allow the water vapor to enter the gas chamber. The water vapor is condensed or cooled, and then the condensed liquid now goes here. There are sources of some small organic molecules here, okay, and then return to the ocean, okay, into the liquid chamber. So what did they discover? After several cycles, okay, several cycles, they were able to get small organic compounds, including amino acids, from this experiment. Okay, so amino acids, if you still remember, are the building blocks of proteins. These amino acids are actually the building blocks of life. So this was the chemical process that they were looking for. They know the physical events. They know the physical characteristic of the primitive Earth. Now this is the chemical process that they were looking for. That yes, it is possible for the conditions of the primitive Earth to give rise to the building blocks of life. After Miller's death in 2007, scientists examining sealed vials from the original experiments. So again, no, this was conducted in 1952, and Miller died in 2007. Of course, they were able to actually preserve a lot of the samples. Now, in 2007, scientists examined the sealed vials from 1952, and they were able to discover over 20 different amino acids produced in Miller's and Urey's experiments. So 20 different amino acids. And again, amino acids are the building blocks of proteins which makes up organisms. That is considerably more than what Miller originally reported in 1952 and more than the 20 that naturally occur in life. So they were able, yes, the Miller-Urey experiment was able to recreate the conditions of the primitive earth as well as the chemical processes to create the building blocks of life. Okay? So, in the 1953, 1952, 1953 study, okay, they also conducted an experiment to simulate a volcanic eruption. Okay, and from 1953 up sometime in 2008, a reanalysis of those results found that amino acids were produced under simulated volcanic conditions. So this is the number of amino acids in 1953. Then in the sealed vials, after how many years in 2008, when they checked and opened it, they were able to find more than 20 amino acids. So during this period, in those sealed vials, the number of amino acids evolved over time. Okay, as well as the mass of amino acids. So from 100 milligrams in 1953 in those seal vials, by 2008, they were over 200 milligrams. Okay. Now, these are more recommendations for you to really visualize how biogenesis happened and how the Miller-Urey experiment attempted to explain the jump from chemical to biological evolution. This one is still from stated clearly what is the RNA world hypothesis. I'll provide the link in the description below. And this one, what is the metabolism first? Hypothesis for the origin of life. Again, linked in the description below. So right after the creation of the first cells, the protocells, okay, life flourished throughout our planet. Okay, but let's take a look. What is a protocell or what is a protobiont? It is a self-organized, endogenously ordered spherical collection of lipids, and this is considered as the stepping stone to our bacterial cells, okay, our primitive cells. So if you still remember the vesicles that we've mentioned, this is one of those possible vesicles that they were able to form their own primitive cell membrane to separate them from the world around them. So these are the three main structures of their structures of phospholipids. Okay, in a liposome bilayer, in a bilayer sheet like this, and in a micelle like this. Now, the central question of evolution is how simple protocells, such as these ones, first arose, and how they could differ in reproductive output, thus enabling the accumulation of biological emergences over time. And we call that biological evolution. We've mentioned this, that life eventually evolved into different forms over the course of billions and millions of years. Now, these are fossils, okay? So this is a cross-section of a fossil bacteria from Australia. So we see here ancient cyanobacteria, and if we see them, they're actually quite similar to this proposed proposed membranes of protobionts. Okay, so this is how their cell membrane or primitive cell membrane possibly look like. Now, this is a microfossil of a primitive eukaryote. This is a multicellular alga between 900 million and 1 billion years old. And this is the oldest microfossil, a bacterial fossil discovered by J. William Spoff of UCLA. That's determined to be around 3.5 billion years old. Okay, found in Western Australia. And this is quite similar to the structure of the present-day cyanobacteria. Now, so we know where life possibly started from simple inorganic molecules, jumping into abiogenesis, then to biogenesis. So now at this point, we have to ask ourselves, so after knowing all of this, the possible origin of life, what is life then? How do we define it? In the previous video, we have identified the characteristics of life. But in this video, stated clearly, it tries to really define what life is. And it's really quite difficult because we have a lot of factors to consider. We have to narrow it down to include all of those organisms in the 3 domains I've shown a while ago. Now watch this video, and this is their working definition of life. This one is actually from NASA. Okay? They define life as a self-sustaining chemical system. Okay? You're familiar with chemical system. We mentioned this. Capable of Darwinian evolution. That is the simplest definition of life. Self-sustaining chemical system. It can create its own cell membrane or membrane to separate it from the world around it, and capable of Darwinian evolution, capable of surviving through natural selection. That is, in the purest essence, the definition of life. Now, to learn more about it, watch this video from Stated Clearly. I'll provide the link in the description below. We've mentioned a while ago that if life could happen relatively easy, okay, relatively easy in our planet, then it is possible that it is also present in other planets, in other systems, in other galaxies. That's the reason why we need to strengthen our space program. Okay, this is an article from National Geographic that tries to justify why we explore Mars and what decades of missions have revealed. I require that you read this article and see why we are putting all our resources into space exploration and what can we get out of it. Okay, now, short answer, the more we learn about other planets, the better we can understand our own. The more we learn about the fates of the other planets, what happened to them, the more we can see the direction our own planet is taking. If you're interested to read this, I'll provide the link in the description below. Now, this video is from NASA. Okay, the Opportunity NASA rover completes Mars mission video. Okay, it's a video that explains to us what happened in Opportunity. That's a NASA Mars rover. Okay, very interesting video. I'll provide the link in the description below. This one also, Curiosity rover snaps billion pixel panorama of Mars. So this one, a very interesting video. You will see that Mars and Earth share a lot of similarities in terms of their landscapes. Okay, again, linked in the description below. That ends our video. I hope you learned something new. Don't forget to subscribe to my channel and like and share this video. Till next time, goodbye.